Oh, welcome to the Cell Project. Um, so I'm now going to figure out how to hold this and draw at the same time. This is going to be awkward. Okay, you know what? Fantastic. Fantastic. Okay, so here, um, why do I suck at drawing? We have a cell. Just so you know, I'm drawing this through like the screen of my phone, so that is why I'm sucking. Okay, arrow. It grew. See? It grew. Bigger cell. Emphasis on that word, bigger. I'm gonna put my phone away then. Okay, I thought they didn't have office. See, now that. Yeah, come on, we got nothing going on. We have sugar and phosphate. Actually. Here we have deoxyribonucleic acid. The sides of it are made up of sugar and phosphate. And then the middle part of it, the rungs, as you can see here, right there, are made of adenine, thymine, cytosine, and guanine. Or guanine, whatever. I don't really care how they're pronounced.
Hey, boys, if you're not going to be grade 9 level about this, then I'm done.
But it can't escape the nucleus. And the ribosomes need it to make protein. So what are they gonna do? Find out! The DNA splits in half and forms RNA. Speaking of, I have a joke. What's a pirate's favorite letter? Ah. See? So here we have our friend ribosome. There are more than one ribosome. There is more than one ribosome, but this one is clearly the best. So he's our friend ribosome. So the RNA leaves the nucleus or the nuclear pore. DNA can't fit through it, but RNA certainly can, and goes to ribosome. The ribosome then reads the RNA, not the DNA, and produces protein, as shown in this nifty little diagram here. The ribosome reads the RNA from the nucleus sends and builds a protein. So we're back here inside the nucleus. where we have DNA once more. But we're ready for DNA replication. Here we have the end goal of DNA replication. But we're not there yet. No, 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 no. As you can see, floating around the nucleus are spare bases. So the DNA unzips. So, the DNA is split up, and stuff is going to happen. Mainly, this might look complicated, but it's really simple if you know what's going on. So, the base thymine is pairing up with adenine, and the base cytosine is pairing up with guanine. And the vice versa is happening for both of these. So that we achieve the end goal of 2 DNAs in one cell. DNA replication for the win! DNA remains in the form of chromatin, loosely coiled, because we still need RNA for the ribosome. For our friend ribosome, still needs to make more protein. And as you can see, for our specific examples, the organelles have duplicated in anticipation for the new cell. And now we enter mitosis. These are the 3 stages of the cell cycle. We just went like this. From interphase to mitosis. I believe I've already explained the structure of DNA, but just in case I haven't. So, DNA looks kind of like a ladder, and it's technically a double helix, but this is DNA, right here. The rungs of the ladder are made of nucleotides, and made of like nitrogen nucleotides, or bases, and the sides are made of sugar and phosphate, whereas I've explained, I believe. But if I haven't, then I'm explaining it now, and otherwise I'm re-explaining it. Evil. We're here in early prophase, and the replicated chromosomes have formed X's, or I guess technically in this case K's, but who really cares? The next thing that happens is that the nucleolus disappears, and the nuclear membrane begins to break down. We need more of a nuclear membrane than that. There we go. There's our nuclear membrane that's beginning to break down. Spindle fibers have begun to form from the centrioles, which in animal cells have migrated to the poles of the cell. Dictionary. 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 It's time for the dictionary. Let's just say that. So we need to define spindle fibers and centrioles. Spindle fibers. Tiny tube-like structures that attach to chromosomes during mitosis. And we have centrioles that are organelles that organize spindle fibers. With the dictionary out of the way, get out of your science textbook. Now let's get going. Gloves, get out of here. I probably should not be recording this. Now we go into... Wait for it. Late prophase. As shown in this diagram, the chromosomes have attached to the spindle fibers at their centromeres, which are the middle of the chromosomes. if you couldn't tell by the diagram that I have drawn very quickly. Now here we have our nuclear membrane that does this. It's gone! Dun dun dun. Even though that's really good. These are the 5 stages of mitosis, and we are moving into metaphase. Which sounds like middle because they both start with M. Metaphase. Middle phase. And as you can see in metaphase, the centrioles and spindle fibers put the chromosomes into a line at the middle or equator of the cell. In anaphase, the spindle fibers begin to shorten, cling, and that turns each pulls the sister chromatids apart into this. Each sister chromatid is now considered to be a chromosome. Telophase, the last stage of mitosis. We now have no spindle fibers, they've disappeared, their job is done. And one set of chromosomes at each end of the cell. Our friends, the nuclear membranes, are back. And so is so are our friends, the nucleoli. The result is 2 nuclei in one cell and they're ready to divide. Now we enter cytokinesis, and we're going to be moving off the whiteboard for this one. They separate into 2 identical cells. Represented by my gloves. They do this because the cell membrane pinches together. And yeah. So that's the end, really. All that's left are the credits. Here we have our credits. Narrator, me. Cameraman, me. Researcher, me. Making adjustment interview. The background noise provided by Annoying Peoples. Thanks, 7, watching.